It is heterochromatic, lots of repeats. Histone H3 does not bind. Histone H3 is usually found where? Where's H3 found? In the nucleosome. Okay. It's not found in the nucleosomes at the centromere. A different, a variant of it, SENPA, is found. The N-terminus of SENPA has a different sequence, okay, amino acid sequence. It actually is what allows the kinetochore to bind to the centromere. So at the centromere, at least in yeast, there is a different histone that is used in the nucleosomes, which is required for binding of that kinetochore. Okay, two techniques I want to talk about. One is in situ hybridization. In situ hybridization is when you take a molecular probe, basically a molecule, that's similar to the molecule you want it to bind to. Okay, so it's going to hybridize, either through hydrogen bonding, usually through hydrogen bonding. Okay, these molecular probes can either be fluorescent, which makes them really pretty, or they can be radioactive. Okay, so you get probe, maybe it's a DNA sequence that's complementary to the DNA sequence you want to target. Okay, molecular probe is radioactive. They hybridize together, put it on X-ray film. You know exactly where your radioactive probe bound now because it's going to expose the X-ray film. Okay, so that's actually what I did when I was in my undergrad. I was using radioactive phosphorus to look for sequences at centromeres in chromosome 21. A newer technique is now used, FISH, fluorescent in situ hybridization. It uses pretty fluorescent probes. Okay? So take molecular probe with a fluorescent label on it, excite it with UV light, and it will fluoresce. Okay? So here's an example. This is a shot of the nucleus, but this is a shot of the chromosomes at metaphase. So here is one molecular probe, which has a fluorescent green label on it. Okay, so a DNA sequence that will bind to a specific region of this chromosome. This probably, I'm not sure, but I'm guessing is the centromere. It's a common way to make sure you know what chromosomes are which. Okay? So you can instantly match up and say, oh, those are both copies of this one chromosome because they have the same sequence in them. Okay. The red is another molecular probe, okay, for another sequence on that chromosome. So these are used often to look for very small deletions or maybe very small insertions or movements of chromosomes. So if the red sequence was deleted from one of those chromosomes, what would you see? You just see the grain. You'd only see one red spot somewhere in there if it was deleted. Okay? Because that molecular probe could not bind there if that sequence was deleted. Yeah? Okay. What if part of this chromosome got moved to another chromosome? What would you see?
Somewhere else.
You'd see red and green not together anymore. Okay, so it's a way to identify big chromosomal changes in individuals.
You saw 3 chromosomes of that? Is that
That would be trisomy. Yep. So
is that also when you can tell them crossing over? Or is that not?
you could potentially. I don't know if it's been used for that. It'd be hard because crossing over happens so fast. But you could potentially use it if you knew the exact, I mean, if you're lucky enough to get the exact moment when crossing over would occur. Okay, and this is the new kinds of karyotypes where they can actually paint the chromosomes. It's the same thing as fish, it's just using many, many more colors, okay? Fluorescent label on a molecular probe, and they call this chromosome painting because you paint the chromosome. Different colors. Last 4 slides. Okay, so we already talked a little bit about this, but that chromatin structure affects transcription. So if there's lots of proteins there compacting the chromosomes, there's going to be less transcription. If there's less protein, the DNA is more accessible, you get more transcription. Okay? So it's a nice choreographed dance on your chromosomes between, are we going to compact this region, or are we going to uncompact this region? Okay. So here's an example. Okay, remember the white-eyed gene in Drosophila? When it's a mutant, what do you produce? The white-eyed gene? White eyes. Wild type. Wild type, okay? It's going to produce red. So here you can see the nucleosomes are nice and spaced out. Okay, regions are open for transcription. You can get transcription of the wild type allele, okay? Getting expression of your nice wild type I. Over here, though, is a heterochromatic region. There have been lots of methyl groups attached to those histones, compacting those histones further, okay? So that the DNA is less accessible to transcription. Now, big chunk of DNA was taken out and inverted, okay? Which we'll talk more about tomorrow. But the end result is now the location of the white-eyed gene is much closer to the heterochromatic region, okay? Well, if it's far enough away from the heterochromatic region, you're going to still have accessible euchromatin. It can be expressed, and you can have red eyes. If, however, the methyl groups were added, because this gene's really close to the heterochromatic region, it will silence transcription. You can't get transcription. But wait, it's a white-eyed wild-type allele. You should have transcription in normal phenotype, right? It's a wild-type allele, right? It's not going to happen because now this gene is shut down. You can't transcribe it. You can't make the protein from it. End up with white eyes. Okay? So this is called position effect. Where genes are located near heterochromatin, if their position changes, their transcription can be shut down and they actually get a mutant phenotype, even though there was no mutation to the DNA sequence whatsoever. Okay? So take away that DNA mutations aren't always the reason you get a mutant phenotype. I know, they want in, don't they? Okay, we'll do X